Hello! Welcome to
Hello.
Microbiology News, where we discuss some of the latest news stories and papers and also decide what paper we're going to go into real depth and detail in next week. I'm
hmm
Fazalam, and this is my co host.
And I'm Danny.
Yeah, we're here to talk microbiology about all things that are great that are small. Um,
Yeah, we want people to participate with us. So if you guys have questions or suggestions for things you want us to cover, send us a tweet at our hashtag. I guess we're on YouTube, right? So you can chat us in the box there too.
yeah. So this has been kind of an interesting week. I guess for in terms of news, coronavirus is spiking again, which is kind of bad news.
Yes. Yeah, all over the world it seems.
Yeah.
Uh, yeah, in my home province of Ontario, they had restaurants open, and now they're not open anymore to try to curb that to curb that spike.
Yeah,
Uh,
we've got similar thing happening in London. We've got like various tiers of lockdowns, and no one really knows exactly what we're supposed to be doing. But we're all supposed to be very frightened, basically, I guess. And
oh.
that's basically the main takeaway we're getting from our lockdowns. That they were shutting down some businesses, some aren't shutting down, and you're gonna have to look through our long website to figure out which is which, and then do some calculations in your head. They assume a lot of the British public.
Yeah, it's complicated to navigate, I think, sometimes all of those different regulatory things. But having trust, I think, in the public health people and trying to follow their best recommendations, that's always the best
Yes.
advice, right?
Yeah, just fingers crossed. But in terms of
Yeah.
papers that have come out like quite recently, so
Hmm.
let's go into get into it.
Yeah.
So let's see. Let me pull this
This
up.
solidarity trial.
Yes, the solidarity trial. So yeah,
You can tell people are like the mood around naming these clinical trials, right? It's just like stick together, we can make it through. Solidarity trial.
yeah. Hold on a minute. The screen share isn't quite coming through. Let me see whether this is coming through. Okay.
Well, I guess I'll start just by saying this is the big WHO trial that I think we've been talking, right? Whenever anyone has said WHO trial, it was referring to this one, right? This is the one that I think they removed hydroxychloroquine and then it came back. Is that
Uh,
correct?
I think I'm not sure. I think so because this is like one of the really big trials. So we did because we talked about
Yeah.
the British version of this, which was the one that found death
Yes. The
methods.
one. Yeah.
Um,
That was being done with the NHS, and that was like an open protocol, right?
yeah.
Like the protocol was being updated as it went.
Whereas this one was a lot bigger. This involved a lot more countries, and
Yeah.
it tested things like remdesivir and hydroxychloroquine. Uh,
hmm
and and I feel like this is the kind of almost the final nail in the coffin for hydroxychloroquine. Uh,
Yeah.
so I mean, there's one graph that I'm gonna jump to quickly that. Uh, I mean, I mean, there's also like the story about remdesivir that I'm gonna go into a little bit later. But the big
Yeah. I mean, it is. It's. Oh yeah.
no, you
Sorry. I
go
was just
for
gonna
it.
say it's interesting to see that they continued their remdesivir trial, and we talked about this when we were going through the remdesivir
Yeah,
stuff. Like, it came. There were two competing trials that were out,
yeah,
right? One that had like very minimal things, and then one that subdivided the groups to find a better result. So it's nice to see another big data point on the graph.
yeah. I mean, the thing. I mean, okay, I'll go to remdesivir now because it's just this trial also doesn't find any kind of effect in remdesivir, but I mean,
hmm
personally, I'm not going to take that as too much of an because this trial was again like the discovery trial. I think it was called in no, I think it was a recovery
Yeah.
trial. Where this is in
Yeah,
very
whichever. The one in China, the Chinese trial.
yeah in here it was basically very severe patients who are who are hospitalized, and that isn't the most advantageous like place for antivirals because
Right,
basically
right.
like antivirals work best on the first like instance where a virus is about to infect, but when the virus has already taken over, they're not going to have that much of an effect. So,
Yeah. Yeah, and that's the same as what they had found in that large Chinese trial. It was just when they did the subdivision right? And then they looked at just early time points. Did they see that small effect?
Yeah. So it isn't looking great, but there's some. So, but hydroxy.
It's in the
Yeah.
toolbox, right? It's in the toolbox for people, but like that toolbox is limited.
Yeah.
And given the fact that there's not a lot of remdesivir in the world
Mm.
and it's kind of an expensive thing, like whether or not we're going to see a big impact from it, like that's, yeah, remains to be seen.
And then hydroxychloroquine, which has had so many negative trials by now, and it's and my favorite thing that meme that has always gone around with hydroxychloroquine is whenever it fails, people go, well, you should use it with zinc, and people going, yeah, hydroxychloroquine with zinc. So when Trump got sick, they didn't give him hydroxychloroquine, they did give him zinc. So the truth is, zinc was the key all along. But I mean, in this one, again, we're seeing some issues with hydroxychloroquine. They say that it isn't a significant, like it doesn't approach significance, but that people who get it tend not to survive as well as people who do get it, which
Mm-hmm.
is something that has always been on people's minds. And there was that big false retracted study where people kind of were suspecting that they'd made up results where they said that hydroxychloroquine was like really severely bad, and then it fell apart because they made up the results potentially. But here this kind of brings that story back a bit, and
Yeah.
and this is
So the mortality is slightly higher.
Yeah.
Yeah.
And so I guess that is a knock against I mean, again, another big was the president, who had spent quite a lot of time promoting hydroxychloroquine and didn't take it. So I feel like, that's the ultimate burn.
Yeah, seriously.
Yeah.
Yeah, they also looked at some other.
Yeah.
Is this a lopinavir and interferons?
Yeah, lopinavir and interferons. And interferons, again, also seem to not have a great effect on survival. Lopinavir, there is a suggestion that it might help, I think, but it's not especially great. But yeah.
What is lopinavir?
It's another antiviral. Let me double check, because I think it might have the same mechanism of action as remdesivir, but don't quote me on that because I could be wrong.
I see. It's another small molecule of some sort. Lopinavir. That's cool. I have my search bar open. I can drop it in. Yeah, some sort of. Yeah, they use it in HIV treatment.
Yeah.
Strange. Protease inhibitor.
Oh, okay. All right.
Protease inhibitor.
So that's the.
Yeah, that's interesting. We haven't. I guess we really have not talked about that class of drugs. It's like cross-class protease inhibitor.
Yeah.
Like a non-specific viral protease inhibitor. Yeah, I mean, we're barely talking about protease inhibitors specific for the coronavirus family.
Yeah.
But yeah.
Yeah, I mean, even the idea of like a broad spectrum protease inhibitor is kind of crazy to me, because all viruses tend to have such very varied. So that is something I probably would want to.
Yeah. And plus, like, we have protease inhibitors, right?
Yeah,
it's not like when you
exactly.
say, like, RNA-dependent RNA polymerase inhibitor, like, we don't have those, so, like, we don't care about inhibiting them. We have proteases. Like, what if there was overlap, right? What does broad spectrum mean in this circumstance?
Yeah, I don't know exactly what the logic. So that's actually quite an interesting question. I have heard that come up, like lopinavir and ritonavir, I think, are things that have been used together. So I think that's something that we might look in. I'll note that down so that I can search for papers focusing on that in the future.
Yeah, just like maybe it'd be nice to learn with the protease stuff. Like that could help us understand a little bit better. Yeah, not just like, there are things that are like not specific. Well, actually, no, it's always. Sometimes there are evolutionary things, right? Like in a protein that makes it like a totally different class. So yeah, it's not crazy. It's just surprising a little bit to me.
Yeah. Okay, so next we're going into vaccines. So this week, a new vaccine candidate was announced. Well, that passed through phase 1/2 trials. So they published a called BBIBP CorV, which is basically another inactivated vaccine candidate.
Yeah.
So I found, I think we did discuss a little bit last week at how it's interesting what what gets promoted where in terms of like the main vaccine candidates. So, so a lot of the in Southeast Asia,
Yeah, because
yeah,
this is another Chinese. Yeah, this is another Asian one,
yeah,
and yeah, it's like in Asia, like the leading Chinese vaccine is an inactivated
yeah.
vaccine, right?
So Sinopharm
Yeah,
and
so it is kind of funny.
yeah, there's like Sinopharm and Sinovac have both got inactivated vaccines. So I think and they're basically the farthest along in terms of like so they published like phase 2 trials already. So, uh,
Yes, yeah.
and
They're in phase 3. They're giving it to the military. I heard that they may also be giving it to like just other large test
yeah, I
populations.
mean, I've heard that some,
Probably as part of.
yeah, that like I've heard like some countries are already buying up these ones, and it's understandable because of all the because while these candidates are kind of the least like fun, at least like innovative,
Hmm.
I mean that
Yeah,
actually is a point in their favor that they're not innovative. They're tried and tested.
sure. Yeah, exactly. Like the manufacturing process is really well known. So like when you see like a confirmatory safety endpoint in like a phase 1 or phase 2, just makes you feel better about the whole thing because like there's so little that's different besides the virus itself, right? The antigen.
Yeah, and it's always been quite interesting to me, like how throughout this entire like kind of outbreak, people have been pumping, say, Moderna, Chadox, all these very
Yeah,
untested platforms that
yeah.
are kind of like I mean almost jumping 2 generations of vaccinology. So they went so
Hmm.
like the next generation when I was hearing was like, oh, adenoviruses, that's going to be a new thing. And then far off,
Yeah.
we'll figure out how to do gene vaccines, but that's gonna be really complicated. Now we're just jumping both, and it's just
Yeah. Right.
it's crazy because I mean,
Yeah. I mean, the world is pretty tech obsessed,
yeah,
right? So, so I'm like not. Yeah, I think I'm jaded enough that I'm not
yeah,
surprised that those things are pushed, even when there's these other great technologies that if we just spent more time understanding them, we might be further, right? But like, no, 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 it's better to disrupt the system and like sell us the newest thing. Again, not to say that Moderna isn't doing well in their trials. I think that they are.
yeah.
And so yeah.
Oh
So
no, don't get
anyways.
me wrong. I really hope that we do get to jump like 2 generations ahead and have like a very
Mhm.
editable, very quickly produced vaccine that we can use
Mhm.
against lots of different like viruses. Uh, but
For sure.
But yeah, I mean, there is room in here for like a tried and tested vaccine that we kind of know how to deploy, and this is something that
Yeah.
comes through in
Yeah.
this vaccine candidate in particular, because the thing that this sets this one apart is they do seem to have gone into a lot of detail into trying to make sure they pick a genetically stable strain, so that when they scale up
Nice.
production, they or the vaccine, so you can see that they're already planning
Yeah.
that in the early
Yeah.
stages.
And again, that's because they know from the history, right? Like polio vaccine
Yeah,
is like a huge inactivated vaccine. They even have live versions. And so like in this space of sort of traditional vaccines, attenuated and inactivated, there's just a lot of thought already right
yeah.
there. Yeah, I don't even know like what does it mean to scale up an adenovirus production, right? Like that one actually maybe has some of the most roadblocks in terms of scale up.
Yeah.
Yeah. I mean, I've also heard the same thing said about like, have we ever made this much RNA for the Moderna one? Is there any, right? Because like, have we just ever made this much RNA in the world? That also might be like a totally new manufacturing
Yeah,
threshold.
I mean, there are so many
Um,
reagents and stuff we have to think about. The chemistry of actually creating them and delivering them, and also the stability of it, because RNA is
yeah,
quite unstable. So trying to ensure
right,
that
right,
that it doesn't like
right.
break down immediately. Because even if it works really
Yeah.
well in like animals or in humans, if the freezer
Hmm.
breaks down, suddenly it's not useful anymore, and you know, if
Uh-huh.
you're putting in the hands of someone who doesn't understand that, then you'll get people who will get coronavirus who had that vaccine, but and won't understand that it was
Yeah,
because of the breakdown in the cold chain, and that will undermine
right,
faith in it.
right. Yeah, totally. I mean, that's like those are all things like basically hopefully in a year from now or like something like that, right? Like we'll be seeing the extent of those problems. And of course, the companies have assured us that they've thought about this, it's taken care of, but like we haven't seen that data, right?
Yeah,
That some of that data we may never see because like manufacturing information like that is not always
yeah.
published.
I mean, for like a company like Moderna, where they've got their like entire like all their eggs in one basket, and if that goes wrong, then they would have spent all that money on infrastructure for nothing, and that'd really suck for them. But then, so
but they are doing it. I've heard that they're making things at risk right now. Like in terms of when we think about pharmaceutical project management, like people are just like that's like the pre-orders are crazy for these companies. But that allows them to take this risk, right? Because they have the pre-orders, they can be like, well, let's just try to build it, right? And hopefully we'll figure out the bugs as we go.
And it helps that the Warp Speed funds basically have given them the space to take those kinds of risks, where they
absolutely.
And I mean, I'm optimistic that if they do get this cold chain in place, if they get all the kind of infrastructure in place, then that will not just help us for today, it'll help us for future vaccines that use the same kind of thing.
Oh, absolutely. Yeah, the promise, I think that's probably one of the big selling points of the Moderna, the new platforms, right? Is like that if it will set us up for the future, even.
Yeah, because there are plenty of other RNA therapies out there other than vaccines that could really be helpful. I know there's like one for Duchenne muscular dystrophy that could really like help use that, or there are also other genetic disorders that can be solved via this, and that's a potentially fascinating thing. So so if it works, and that's if it works, then it's fine. But
just before we leave the topic of this vaccine, I just want to plug too that like it's funny to think about like this vaccine methodology, delivering RNA, like making our cells express things. It's also like a bridge to gene therapy. Like you just said, this muscular dystrophy, right? That's a genetic disease. And so, I mean, there's still a lot to talk about in the public discourse about gene therapy, but we won't talk about that now.
No, I think it's important. But the big vaccine candidates are basically gene therapies that have been reskinned as vaccines, and almost like, I guess I can. There's like a stealthy.
There's a lot of unanswered. There's a lot of unanswered public discourse that, like, I feel hasn't happened yet, like about gene therapy. Like, we have our favorite Gattaca moments or something like that, like our favorite thing to point at, but like sort of the more nuanced discussion of it, I think still isn't quite being discussed. Like, have you talked to your friends and family about if they would get a gene therapy?
Yeah, I mean,
do they know the difference between germline modification and not? Like, I think these are questions that we should be asking along the way.
I mean, are you... Like, I mean, if like they have a cure for deafness or something like that can actually have some real cultural implications, because you could extinguish languages because of that cure. I mean, that's so there are definite like real interesting things that are raised by this, and almost like, but the thing is, we don't have the technology, so because we never have the technology, these sorts of conversations would never happen. But if these next generation vaccines go through, then those conversations will start needing to be had. And
for sure.
I feel like that's like another reason.
The urgency goes up a little bit more to talk about it.
And that's I think another reason why these perfectly like reasonable like heat-killed like inactivated vaccines aren't being talked about as much, because they don't have that same gateway of technology to the future that these.
Yeah, totally.
Even though I'm reasonably sure that these will work fine and actually might like be the things that. So like
I don't see why they would work worse. Like, first of all, especially until we see based on all this phase 2 stuff we're seeing, they all make neutralizing antibodies, right? So it's like in the comparison sphere, to me, they're all super viable. Hey, let's get it out. Let's do those phase 3s and let's start seeing the long-term effects on the population.
I mean, only the adenovirus-based vaccines have been halted due to random like side effects. So.
Right.
I mean, a great
sort of evidence that our systems are working and we should be doing all those phase trials.
Oh yeah, no, definitely. I mean, I think for adenoviruses, they are extra sensitive because adenoviruses don't have a perfect safety record. So if someone sneezes wrong or it makes, they're going to be like, nope, we're holding the trial. We need to make sure, because there's that extra like wariness about that.
Totally. And actually, I think that's a good segue into this BANCOVID.
Yes.
So like trying to think about making a vaccine against this variant, which I mean, we haven't talked about.
Yes.
Right? Like the D614. A few of them came up 2 weeks ago as something. I remember I had a paper about like the functional difference between this variant and another coronavirus.
Yeah, so this D614G variant that like arose like in Wuhan and was like the founder for a lot of like European strains of coronavirus. So it's spread along the world. So there's been this question of, is it super transmissible, or is it just because it was like the first one out of China, and because of that it's just end up being dominated. So it's so trying to answer that question has been quite interesting because this one,
and in the paper that we covered last week, they did cover this, right? They made sure that their antibody cocktail hit the variant.
Yeah, definitely. So it's something that people are definitely concerned about. So this variant has an aspartame at like that's been changed to a glycine at a certain point in it. So it's, and people are trying to plan their treatments for this. And so I spotted this paper, which is from Bangladesh, where they out of nowhere tried to create a mRNA-based vaccine based on this strain of D614G. So there's mutated in the spike protein, and it's kind of interesting that they jumped towards this. I mean, I had a brief look through the
I think they're trying to stand out by saying that they're looking at this mutation, but from my understanding at least, the mutation doesn't have that much to do with the type of antibodies that will be raised to it, right? So even if you don't have the mutation, you're going to have antibodies against the spike.
Yeah, I think because I think the main concern was whether there was like an immune difference rather than like a structural difference, because like the idea, because if it does increase like transmissibility, then it could be because the structure is more wobbly, so it can grab onto things more, or it can be cleaved by different metastases or something.
Sure.
Or like it can mess with the immune system, and we're worried if it can mess with the immune system, then it will mess with our vaccines. So I guess. So I mean, this paper is very like much we made something, we put into mice, it created antibodies, blah. But the next paper is quite interesting, because this also focuses on the D614G. Where did I put that?
Yeah. And I think it's trying to allay concerns, basically, right? It's like, is there worries that this might not work? But they tried to they don't have sort of bench work about this, but they have in silico evidence to try to...
Experimental and in silico evidence suggests that vaccines are unlikely to be affected by D614G mutation in SARS-CoV-2 spike protein. And I particularly like this paper because it shows the return of Inovio to the conversation, because they use the...
your favorite vaccine?
My least favorite vaccine. So this time they're electrocuting ferrets, and with various isolates with I think so they basically injected them with the Inovio COVID vaccine, then tested out some virus isolates that had this D614G mutation, and and they
and it doesn't seem to have a big
it doesn't have a big effect. They got some nice like modeling stuff on there, some really nice pictures.
So wait, so they do wet work? I thought it was just in silico. Based on the title, it's just like,
they do a bit of both. So they
well, that's nice.
Because I mean,
I see. It's kind of like they have the experimental model where it's like neutralizing antibody titers aren't so different, but then they map that to like the structure, and they're like, well, the structure probably isn't going to change that much.
I mean, I think the main takeaway is that the actual D614 mutation is really nestled inside of the virus. So it's hard to get, and it might not be have any effect for structure at all. I mean I think there's this one like one analogy someone did was like there's this thing that happened during World War II where planes got shot down and they had to figure out where to put the armor, and they put the armor over where the holes were. But obviously that's so I think the idea is that this mutation is happening here because it's the only place where any mutation happened that won't mess with the whole spike protein.
Gotcha.
So
okay, cool.
This is quite an interesting one. I mean, it's good to see that Inovio is around.
Yep. Okay, let's see. We have antibody-like proteins that capture and neutralize SARS-CoV-2.
Yeah, so this is basically following on from antibodies, but they've synthesized their own antibody-like proteins, which
Yeah. Yes. What I'm really excited about this one is we've never talked about phage display.
Yes.
This is one of my favorite biotech, like recent. I guess not recent. It's been around for a while. But biotech techniques using viruses and directed evolution really to get closer to something that you want. We talked about the germinal centers and how our bodies use like a type of directed evolution, right? To like make sure they get the best antibody out. And so we don't do it with cells because that's really complicated. We are more advanced with phage, and so we actually do it with viruses.
Yeah. So the interesting thing is the technology that's used here, and it's it's quite interesting, and like just looking at the kind of because I think this is very much up in the air whether this will actually work well in the lab, but it's interesting to see how this sorts of thing are developed, and because to me it basically sounds like a molecular machine that they've just constructed that's based on antibodies, but we trying to be making things more efficient. Like
totally. I mean, this is like real. To me, this is like a lot about what synthetic biology means, right? Like what the hell is synthetic biology? Well, to me, it's these techniques really feed into that. It's like, we're just making things like they're biological molecules, but they don't really, like they're based on something we saw in nature, but we've taken it into our own and like really crafted this molecule in our own way.
Yeah, I mean, I think that's something that's going to be more important as years go on, because the applying engineering principles to biology is something that is becoming something that less of a pipe dream than it was a few years ago, because
absolutely.
So that's what interested me, and because the future is thinking just.
I mean, when we're talking about all this, mRNA, lipid nanoparticle, right, being the future of this, like here's another type of frontier, maybe even earlier in some ways, because like as far as I know, people just use this. People use this a lot in the research and development phases, right? And so I'm not sure people. It'd be nice to speak, but just to talk about the technique.
Yeah.
Okay, let's see.
SARS-CoV-2 disrupts splicing, translation, and protein trafficking to suppress host defenses. So this is another very molecular kind of paper, but it talks about all the other proteins of SARS that aren't spike, which brings it up in my book, because those are the things that we really don't know too much about.
Yeah, all the obsession with making the spike vaccine, right? It got us to the point, right? Like, it's almost like, all this basic research, there's all these different things. One kind of emerged to get application, and now slowly everything else is kind of filling in as well. And we're seeing, the bar of knowledge on everything else rising.
Yeah, and I think that's a really good thing. And it just, it's something that I find fascinating as well, because the way that viruses do things can be very, clever and because they're very...
Absolutely. They're such minimal, I was going to call it organisms, but not organisms. Yeah. Minimal self-replicating particles.
Yeah, they're angry genetic code. It's so they're like bullets. They're so unique and interesting that way.
I think this is a little bit of a sidebar, but I think fun to mention. It reminds me that computer viruses, they come second, right? It's not that we named the viruses after viruses. It's that computer viruses were named after the viruses, OG, in the world, right? On all the wetware and different operating systems that exist, there exist viruses.
Yeah. I think that's... Even viruses have viruses. I mean,
even viruses have viruses. Yeah, exactly.
Because it gets to the point where, it was this bits of genetic code arguing with each other and trying to sneak away with as much, ability to replicate themselves as possible. And
exactly. They wanted to be the last thing written on the shape of life on Earth.
Yeah. There's this theory that they were the first thing written, but in the beginning we just had bits of genetic, whatever the version of genetics would look like back then RNA, DNA, whatever, all of them just come together, and this is why I like microbiology, because it is looking at life at its most primitive and most kind
Yeah.
of, you almost see like kind of the blueprint for everything in all parts of microbiology. So when this coronavirus is outbreak is finished, we might get to the chance to talk more about that. About like, yeah,
Absolutely. But I mean, I think this paper is a good bridge because this is sort of exactly where you would get that insight into life by studying a virus, right? Because we're looking at the specific mechanisms our favorite, our infamous virus is using to deal with host cells. And in the details of that, we might find things that are surprising and tell us more about just life in general or the way biology works.
Yeah. And that's always
because these are big, like splicing, translation, and protein trafficking. Those are like big concepts in cell biology.
Yeah, I mean,
yeah, those are big processes.
Yeah, those, I mean, without translation and protein trafficking, you don't get life. So, and so how is SARS-CoV-2 disrupting these specifically in the host cells without killing the host cells off outright? Because they still want to replicate their own stuff, and this also ties back into that replication organelle we were talking about a couple of weeks ago. So it'd be quite interesting to see whether the work here reflects on that.
Yeah, and I think maybe it's no surprise. This is like probably one of my favorites in this list. The other thing to say is that splicing also hits like another interesting topic because those proteins are related to all proteins that like bind DNA, have to recognize a sequence and like cut and repaste things, right? And like that's a big area of sort of emerging biotech comes from those types of proteins, right? CRISPR is a similar type of, like, at the general sense, that similar type of protein.
Yeah, no, I remember, like, I was fascinated, because, like, about 10 years ago, I was at this conference, and I was fascinated about, like, because that was before we knew what CRISPR was, but I knew it was messing with my experiments, because I was trying to genetically modify bacteria, and I knew that CRISPR was one of the things that shouldn't, and I was quite interested about it, and so I ended up talking to this lady who was very into it, called Emmanuelle Charpentier. I was like, oh, wow. It's like, oh, it was one of those moments where it was like, I think my supervisor was really pushing me to go to a lab. I was like, I don't know about this. I am kicking myself today. I am because I had a long conversation about CRISPR, and she was trying to explain why it was so important. I just wasn't getting it. 10 years later, oh shit.
That's why, yeah. So splicing is in that area, and
splicing is very important.
Knowing how those proteins work better could unlock sort of those types of manipulations.
Don't make my mistake. Don't sleep on splicing, kids. I mean, that was my error. I just thought, oh, this is a minor thing. No, it's going to be really important.
Well, hindsight is 20/20, right?
The whole year is 2020.
The whole year is 2020. It's always it has been the whole time. 2020.
Yeah, 2020. This is a year where we reflect on all of our mistakes. It makes sense now. Now that I look at it from 2020.
Okay.
Oh, sorry. We'll go back to real
okay, we got architecture of inactivated SARS-CoV-2 with postfusion spikes revealed by cryo-EM and cryo-ET.
Yes, cryo-EM, favorite technique now.
Our favorite technique, just like a fascinating technique for looking at tiny objects, no longer relying on crystals. We could see them frozen in weird jelly ice, glass-like ice.
The thing I like about this is they use beta-propiolactone inactivated SARS-CoV-2 viruses. So those are basically all the inactivated viruses that all of them use beta-propiolactone to inactivate them. Yeah. So this is quite interesting from the perspective of like those vaccines we're talking about.
Sure, it gives us some insight into what does the vaccine even look like that we're injecting into us, right?
And I think that's very interesting because you'd expect them to all like just be in the spike in its pure state rather than in a post-fusion state.
The pre-fusion state. But somewhere in the post-fusion. Yeah, that's a really interesting observation. It makes me think about these proteins like they're it's a term that's used. They talk about with injection mechanisms in bacteria, right? Like bistable or something.
Yeah.
Like they have like an energy state where it's like they could go either way.
Yeah, they're like,
right?
like a cold spring where they're just ready, they're held in a specific, and the moment something happens, they spring up and they turn into the postfusion conformation.
Yeah, it just happens. And like that energy of the transformation is what's useful. Like if they're in the right spot and that energy is released, that's where you get the fusion, right? But like using this inactivating compound, basically, I guess it tickles them in a certain way, where they shoot it off and then they're frozen in time, and then we can look at it with powerful electron microscopes.
I mean, it's basically acts like glue to like make them more sticky and weird. So maybe that's what triggers them. I mean, it's fascinating to think about, and it might have implications for vaccinology. So
I didn't think about that, but that is interesting about this. We're learning both about like the structural biology of like the mechanism of this protein, how it gets from stage because we're seeing the stage before it fuses and the stage after. We have like snapshots, right? And scientists can try to like imagine the animation that would lead to that. But as you said, it also gives us insight where we're like characterizing actual vaccine components. Which could also have implications on the safety design, or like at least interpreting safety information as it comes out.
I mean, even interpreting like kind of how well it works, because I mean, we could say like maybe if they can control the amount of prefusion and postfusion, you get a better vaccine, right? Like this could be developing the inactivating vaccine to be better based on this sort of information. Yeah. Okay. Next one is differential expression of viral transcripts from single-cell RNA sequencing of moderate and severe COVID-19 patients and its implications for case severity.
A big omics. It's an omics one.
Big omics. We haven't done much omics.
We haven't done too many omics. Here's another in the list. This time it's being used to correlate. So, the classic things with omics papers, there's no causality is a really hard thing to figure out in this case. But here they're looking at the signature, right, of what the viruses are making inside of sick people, and saying does that correlate? And then if you can get correlations from those things that are being made, then you can start asking what might be what might be the reason, and can we start verifying some of those things with more mechanistic style studies?
Right. So because it seems like they like do some characterization of like about what's happening to the host cells, and it's I mean it's.
I think this is based on I think maybe 2 weeks ago I was interested in seeing they did like a mass spec omics where they looked at like all the different proteins that are being made by SARS-CoV-2 inside of a cell culture infection, and like I almost think of this as like this study can follow up on that, right? And say, now that we know all the little things, like in an actual infection, what do you actually make? Right? Or what's likely to be made there? And then again, like, there's like so many different ways of getting at a question. Omics has always been the top-down way, where it's like you see everything, and now you're like, the challenge is what's important in the image that you
there's
saw.
like always a ton of noise, and trying to figure out what's like signal and what's noise is a big like kind of. And also what to focus down on too, because sometimes you have tons and tons of work ahead of you, and you only have funding for like one section of it. So just all that prioritizing.
Totally. Yeah. But they do find some sort of signal in this. And so that's basically all you can say from some of this. But reading it, we can get an understanding of the methodology of taking these big data approaches. They're very important in science now, but not the only thing that goes into making good science. I think that's important for people to remember as we become obsessed with large data sets.
And I guess the final paper we'd be looking at is the superantigenic character of an insert unique to SARS-CoV-2 spike supported by a skewed TCR repertoire in patients with hyperinflammation. Now, superantigens are something that I've been really interested in during my PhD. There's this antigen that can non-specifically just cause all the your immune system to react to it. So it seems to be
the classic superantigen is toxic shock syndrome, right? Yeah, or at least that's where I first heard about it, was with it's a famous case in, I think, in the U.S. Correct me if I'm wrong. I think it was a U.S. case where, leaving your tampon, people
Yeah.
were observing this thing where, like, if you left your tampons in for too long, people came up with this, like, really crazy disease, like a cytokine storm, like immune levels elevated, and no one knew what it was. And it was it was Staph aureus, a bacteria that lives on our skin that creates a superantigen in the specific environment of the tampon.
That, like, binds antibodies and causes your immune system to react to everything. And it's so the one I know you worked with staph, I worked with strep, and they have different.
What's the strep superantigen?
So strep also has super antigens that also do very, like, similar things as, like, bamboozling the immune system, and it's something that I never really thought of for viruses for doing, but obviously, like, it'd be quite interesting, especially since we did see something like suggestion that there is an unbalanced immune response to SARS-CoV-2. And
totally, I mean, like. The way that people have been describing that unbalanced immune response, at least from what I've been reading, right, is that it's that, like, lung infiltrate, right? It's like, it's just, like, super active. But, like, why is it super active, right? We never really went down. Is it because it's replicating really fast, right? Is it like, who knows? But the suggestion here is that there could be some evolution inside of SARS-CoV-2, right, that has this very specific type of a very specific. I don't know what is a super antigen? Is it a motif?
So,
no, it's like a functional definition.
It's more of a functional definition. So usually there's something that is either, like, very, like, kind of. So I know for staph they have a super antigen that binds, like, just directly to the antibody and to, like, a cell receptor to make it.
Yeah. I think that's the functional definition, right? It's like it's just something that can activate previously antigen-specific immune responses and make them, like, not specific, just, like, activate the immune system.
Yeah, so where your receptors wouldn't bind, this superantigen comes in and forces them to bind, and then suddenly your T cells think, oh no, I need to destroy this cell now, even though it's completely innocuous. I mean, there's sometimes...
Exactly.
And I know there's some versions where they look very similar to host cells. So, like, where they...
Well, I mean, it can explain... Superantigens sometimes can explain, like, different types of autoimmune sequelae, right? Things that happen after the infection. If you have autoimmune things, maybe it was, like, something that came out in a whole superantigen mess, where it's like your body recognized the wrong things and directed it down a non-productive immune response. It's an immune response, but it's not productive in the way that you need it to be.
This is mainly a computational modeling paper, so they don't have as much, like, they don't really seem to, like, demonstrate much in vivo. So
it's still kind of a hypothesis of a superantigen, I would say.
Yeah.
Right? That's like, this is the title is giving us a bit more. Yeah, I thought this too. I was like, if it's a functional definition for superantigen, then should we be seeing some functional data?
Yeah, that's kind of what we're hoping for. So this one kind of clickbaited me into it a bit. So, I mean,
I think their main line of evidence, I think, is the TCR response, is how they're saying it, right? So maybe that's something. I didn't think about that, like repertoire. We were talking about that last week, right? Because if the repertoire that you see is equally distributed, right, over all these different ones, then you know something is activating it across everything, right? So that could be, I think, what they're saying here. Okay, and it's not so clickbaity, but it's a little bit clickbaity. Like, we'd like to see, like, actual superantigenic responses.
I think we'd really want to see, because, I mean, with lots of proteins, there are ways you can try and make them look like they match things that they aren't, which is why often protein research has lots of kind of safeguards in place to try and make sure models work in a specific shape. So.
Oh, but I'm saying that it does give us one piece of data that isn't just shape match, right? It gives us the repertoire information, and that's different.
And that's quite interesting.
Okay, so that's a good load of things. As I said when we talked about it, I'm most interested in the Banerjee one, the disrupt splicing, translation, and protein trafficking.
I'm quite interested in that as well. So it was a choice between that or the antibody-like proteins that capture. Those are the
antibody-like proteins. Those are the meatier ones. Yeah, let's look at when you open up antibody-like proteins.
So, I mean, but the thing is, antibody-like proteins, that is more about their technology, whereas this is actually about the SARS-CoV-2 virus. And
Mhm.
and so yeah, I mean, when I was selecting this, I thought that this was going to be one of the winners because it covers quite a lot of interesting things that we haven't really covered before, and things we wanted to look at,
Absolutely.
like such as what the non-structural proteins are doing, and
Hmm. Hmm.
and looking
Yeah,
at
I'm looking at the figures, just like scrolling through, right? Like this is we've been trying to figure out, right? Like this is why we do this off week thing. Like how do you choose something to zoom in? Because
Yeah.
there's like so much information out there. Just like scrolling through the figures is sometimes nice to get a sense. Oh yeah, they have structural pictures, they have Western blots, or they have something blot. Oh, it's intense. This energy one. There's also like techniques and things in here I think that we haven't talked about as well, like basic biochemistry techniques potentially.
yeah.
I can't tell per se, right? Because there must be. I think there's some sort of translation test that they're using to make sure, right, to prove that it's blocking these things.
Yeah. So it's quite a comprehensive set of data,
Yeah,
and
it's a big
yeah,
7 figures. That's a I mean classic cell paper.
yeah,
Right?
classic. I mean, I feel like I've heard of some of these people before, but I can't quite think about it. But I know that yeah, it's sorry, the actual PDF is taking like quite a long time for me to download to show on stream. It is a very meaty paper.
Okay.
Yeah,
I'm up for that. I think I prefer this one over the antibody one. The phage display. As much as I love phage display, like, I think that I do want to know more about the basic biology of this virus.
yeah.
And it's like such a great time.
I feel like if we don't read it now, it'll get lost in the wash of only more information like this will be coming out.
Yeah, and with phage display, that's a technology that's out there, and we will see more of it. So I don't.
Yeah, I mean, hopefully, maybe even after SARS-CoV-2 is over, we'll be able to talk about phage display. It'll be the next best thing.
Whereas, like in 5 years, people won't be. Hopefully, I hope people won't be googling SARS-CoV-2. That's me being optimistic.
This will just be another virus in the annals of what are other viruses in there like Rinderpest.
Yeah.
Rinderpest. You know that? Do you know that virus?
I mean, does that affect cows only, or is
it's a cow. Yeah, but it was a huge outbreak of cow. They had to slaughter so many cows back in the day. But we don't talk about that one anymore.
Yeah, I mean, even like MERS, we don't talk about as much, even though it's relevant. We just don't talk about it.
Absolutely.
But
yeah. Well, in some ways, we probably should have been talking about it more, right? Like in hindsight again.
We did our due diligence on that. We did talk about the MERS spike protein a bit, and so we definitely. So I.
Oh yeah, no, us as a group. I'm saying us as a society, the obsession, the sort of flash in the pan that MERS and SARS, they were relevant only when they were affecting people's human health, but the dialogue of what does it mean that these viruses are emerging, right? That kind of broke down a little bit as it went on.
Because
but we have talked about MERS.
Yeah, we have. We've done our responsibility. It's society's problem. I take no responsibility about the. But
how are you feeling? Do you think the Banerjee one is?
Yeah, I think the Banerjee one's a good one to look into. I feel that's something that it's one of those papers that will feed into our understanding of other papers going forward. So I think that
absolutely, as we try to build out our knowledge of how does this virus work at a mechanistic level as a way of describing, it only helps us talk about vaccines better, right? Because it sort of fleshes out our knowledge of what are the other ways the viruses might be getting around. So God forbid, we don't see vaccines that don't work, right? But if we get to that point, we can then take that knowledge and take it a step further, right? And be maybe it's because of this, right? Maybe if we inhibit this thing, then our vaccine will be better.
So I think that covers it. I think it's been quite nice talking to you about this, and quite nice chatting about catching up on the news and everything. So,
absolutely. So yeah, for people who are listening on, if you guys are going to read this paper along with us during the week, then please send us your questions or your comments, I guess, your impressions using the hashtag micro Twitter journal. Micro TWJC, and we'll be back next week and dive into this paper figure by figure.
Yeah, thank you so much for joining me, Danny. It's been great talking to you, and I'll see you.
Talking to you.
Yeah, I'll see you next week.
Okay.